Okay, so last time we started looking at the types of structures that are generated by proteins. And particularly, we looked at secondary structures, which would be the alpha helix, the beta sheet, the zinc finger. Those are the secondary structures that then come together into larger complexes, more complex 3D structures, which would be a tertiary structure. So it's more complex, multiple secondary structures coming together, and then we kind of illustrated by these ribbon diagrams that I had talked about, and I said that they are generated from experimental data. So I want to show you a little bit of how that's done. So in terms of proteins, there are believed to be well over 150,000 different proteins in our body. There may be as many as 1,000,000, maybe more. It's not quite clear. They all have very specific structures, and each structure has a different function. The function is totally dependent on the structure of this thing. And some of them look something like this. They're very, very beautiful in a sense, if this is what they look like. But you can see lots of alpha helices, lots of beta sheets, forming this large complex, which has to look exactly like this in order for it to function correctly. They're much more complicated than nucleic acids, and that's why I said at the beginning that we'll be talking about proteins for a little bit longer than nucleic acids. Nucleic acids, you have double helix, or you have some other types of simple structures, but nothing like this. So the question is, how do we come up with something that looks like that? We come up with it using data generated by what's called X-ray crystallography. So I want to talk a little bit about this technology. The X-ray crystallography is used to determine protein structures. And I'll give you a little bit of history as well. So you can take a purified protein. So if I want to work with, an antibody, you can purify that antibody, and you can crystallize it. It has to be extremely pure, and then you slowly dehydrate it in a test tube or in a small dish. And what happens is it forms a crystal-like structure. And when it forms a crystal-like structure, all the protein molecules are aligning in such a way that all the similar structures somehow are lined up together. It forms a crystal. And this is an example of a protein crystal. Now, in a protein crystal, the atoms are aligned. So they're oriented in some particular way, so the atoms are aligned. And what you do is you hit these crystals with X-rays. And the machines, the X-ray machines, look something like this. This is an X-ray crystallography machine. They cost over $1 million. They're extremely expensive. And you can see, it's hard to see, but right in the middle there's like a probe with a small, right in the center, right where the two glass plates kind of come together, the protein goes on the tip of this little holder. And then there's X-ray that's generated and shot through the crystal. And what happens is when the X-ray is shot through the crystal, the atoms that are aligned together cause the X-rays that are coming through to be diffracted. That means they get deflected in a particular way. So here's kind of what it looks like. So you have your protein crystal shown in B, the X-ray source, crystal is put in there, and then you shine the X-ray through the crystal, and then you get this, the beams get diffracted by the position of the different atoms. and then it's picked up on a digital detector. In the old days, they used to use film. I'll show you in a second. They used to use X-ray film, but now they're picked up on a digital detector, and then that information is fed into computers. So, does anybody know what this is? Again, if we were in class, I'd wait for an answer, but I'm not going to get any answer, but some of you may know. This is a structure, this is an X-ray image of DNA, of double-stranded DNA. This is the image generated by Rosalind Franklin in the 1950s that Watson and Crick used to determine the structure of double-stranded DNA. Now, if you look at the picture, so X-ray crystallography was developed in I think the 1940s, and they started using it in the 1950s, and they used it particularly to look at nucleic acids and also other elements and other small molecules. But if you look at this, you can see it's, we know that the DNA, we'll talk about it later, DNA is helical, and you can see there's symmetry there. If you look in the middle, there's those four large black spots, kind of long dashes that are present 4 times. Everything is repeated, so it's symmetrical, and it looks spherical, and they were able to measure just the distance between those different spots, and then using those distances calculate the distances between the different bases and the turn of the helix and so on. Now, proteins are much more complicated, and people tried to use X-ray crystallography, and they'd get these images, but it was just way too complicated. So X-ray crystallography stopped being used from, I would say, the early 1970s until recently, with the advent of supercomputers, computers like what we looked at earlier, but they don't have to be that sophisticated, but you do need to have computers that are extremely fast and can handle a large amount of data, because the X-ray crystallographic images of proteins, rather than looking like this simple one that you get from a helix, DNA double helix, they look something like this. And all these spots and all these patterns represent the structure of the protein. Now, nobody can sit down and measure the distances between these things and the arrangements and come up with any kind of structure. It's impossible. So what people have done is computer scientists have developed algorithms that would generate structures by analyzing the patterns of these X-ray crystallographic images. So it takes high-speed supercomputers, it takes very sophisticated programming and algorithms to do this. And so once computers became more useful, the X-ray crystallography became more popular. And you can see here, the use of X-ray crystallography has increased dramatically. So if you look at so there's a number of entries into database, basically a literature database, looking at the number of papers that have published using X-ray crystallography. And so what you can see is on the y-axis you have number of entries. In 1976, as I mentioned, nobody was using it. It just couldn't be used. They were trying to look at proteins, but it was too hard. And then as you see, as we start getting into the 1990s, 2000s, and of course now 2011, 2017, 18, and so on, there's been a dramatic increase in the use because the computers have gotten much more sophisticated and much faster in order to be able to handle all the data. And so that's the blue is total number of papers that have been published, and then the orange shows the total number of protein structures that have been analyzed or determined. So if you were to add it all up, it looks like maybe we'd have something like 20,000 different proteins, structures that have been determined using X-ray crystallography. And of course, there may be as many as 1 million or more, so it's just a very small, relatively very small number. So we end up getting structures that look like this. This is the ATP synthase. This was the protein that allows the protons to flow through and generates ATP. So you get these structures, and we think that's kind of what these things look like. Here's another protein. This one is a protein called catalase. It's involved in breaking down hydrogen peroxide. Again, completely different structure, but generated by X-ray crystallography. Here's another very pretty one. This is the alpha hemolysin. This is a protein that's generated by bacteria. It's a toxic bacteria that makes a pore, that integrates into the cell membrane and makes a pore in the cell and basically destroys the cell. You can see lots of alpha helical structure and this kind of circular shape. And let me show you one more. This is from the cover of a journal called Cell, and what it says, you can see on the structure on the cover of the journal. It says crystal structure of sliding DNA clamp, and we'll talk about this clamp, otherwise known as pCNA. It's involved in DNA synthesis, and we'll talk about that when we talk about DNA replication. But what I want to show you about this thing here is that in this case, what happened was they co-purified DNA and the sliding clamp protein. So they bound the clamp protein to DNA, purified that, crystallized them together, and then carried out the X-ray crystallography, and the computer generated this picture. And what's remarkable, at least to me, is that it really shows the sophistication here, because what we're looking at the center is kind of a cross-section of the DNA with the proteins surrounding it, making a kind of a clamp on the DNA. What you don't see are the bonds between the DNA and the proteins. Looks like the alpha helix is closest to the DNA. You don't see the bonds between the different proteins. But I will tell you that we know this is pretty accurate, because if, for example, you have a figure that looks something like this, and then you make specific mutations in the protein. Let's say it looks like from the image that a tyrosine, let's say a particular tyrosine, looks like it's making a contact with DNA from the X-ray images. So you go back to the lab, make mutations in the protein, so you knock out the tyrosine, so there's no tyrosine in there, and then when you try and bind the protein to DNA, it doesn't bind. So we think that they actually kind of look like this. Now, in addition to the X-ray crystallography, we can also use electron microscopy. With electron microscopy, you can visualize things, atoms or molecules that are extremely tiny. So the best light microscope can magnify something maybe around 1,000 times. But the best electron microscope can magnify something as much as 5 million times. 5 million. And if you do some simple calculations, of ask about the size of a cell and what 5 million times would look like, so in principle, you can take a cell, a single cell, and magnify it to the size of an Olympic swimming pool. in focus. So you can see the organelles. You can see in detail ribosomes. You can see in detail mitochondria. You can see microtubules and all that stuff. So it's very, very powerful, and you can see proteins. You can see DNA. So we can visualize proteins. Now here is a X-ray crystal structure of myosin. So myosin is a molecule that's involved in muscle contraction. And what people did here is, of course, they purified myosin, they crystallized it, they did the X-ray crystallography, they generated the image. The computer then used that image and the data points to generate this structure. And if you look at the structure, there's long alpha helical section in red, and then there's kind of a more globular shape on top in green and blue. So this is what the X-ray crystallography tells us that this protein looks like. So we then go ahead and kind of make a drawing. So then we take that X-ray crystallography data and the image that came out, and we draw something like this, which is, like what you'd see in a textbook. So you have the alpha helical form, the alpha helical region of the protein. Actually, there's two alpha helices. They're bound together. They're amphipathic alpha helices, so they coil together to hold these two proteins together. And then at the end, like what was shown, I think it was in blue, I don't remember the color, no, green, you get the more globular structure. So this is what we get from combining the X-ray crystallography data and then what we think happens when you put these proteins together. And now if you purify myosin and you look at it under the electron microscope, you get something that looks like this. So the X-ray crystallography and the drawing that we made is what the protein looks like. This is it. This is a molecule of myosin. You can see the long region, which would be alpha helical, the amphipathic alpha helix, and then you have the more globular groove up on the right-hand side. So my point is twofold. One is that the X-ray crystallography is pretty good at representing what proteins actually look like. And by electron microscopes, you can actually see that these proteins look like that. So these are real structures, small little entities made up by molecules, and they carry out these activities in cells. Okay, so we're going to stop at this one, and we're going to talk a little bit more. I told you there's a lot to talk about. At least for me, I feel there's a lot to talk about. We're going to talk about tertiary structure in protein domains, but that's going to come next. Okay.